This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs.
So my name is Aimee Kao, and I'm an assistant professor here at UCSF in the Department of Neurology. Along with my colleagues, Dr. Dina Dubal and Dr. Jen Yokoyama, we're going to be presenting tonight on the topic of genes in the brain from worms to people. And we'll each give a little introduction about ourselves prior to our section. I didn't actually plan an introduction. I just learned that we have to give an introduction. My bio is actually in the packet, I think, that you have, and so you can certainly read that. I guess the one thing that is of note and that I'm proud of is that I've done basically in my entire postgraduate training here at UCSF. I was first a neurology resident and then a fellow in the Memory and Aging Center. Then I did a postdoctoral fellowship with Dr. Cynthia Kenyon here at UCSF. And last fall, I believe, I started my own group studying neurodegenerative disease. And I'll tell you a little bit about the work that we do as it relates to this topic that we're covering tonight. So this is, I believe, the fourth session in this mini med school course. And looking over some of the slides of the presenters over the last few weeks, it looks as though you guys have already covered a lot of ground. So first you heard about the brain and creativity from Doctors Miller, Viscontis, and Vossel. And then you learned about some specific diseases, Alzheimer's disease and FTD, as well as the neural networks that control them, as well as drug trials that are ongoing to try and attack some of these diseases from Doctors Seeley, Rabinovici, and Boxer. And then 2 weeks ago, you learned about some of the tools of the trade, some of the neuropsychological tests that we use to kind of define the brain, to learn about these diseases, and also to train the brain. And then it sounded like you had a really interesting session with Dr. Chung about learning about what the brain is in relationship to the self. So we're going to take a little bit of a different approach to the brain tonight, in the sense that we're not going to treat it as some sort of mystical creative force, which it is, or a complex series of neural networks, which it is. We're really just going to think of the brain in a very reductionist way, in the sense that we're just going to think of the brain, I think, as simply the expression or the product of our genes. We really are simply a reflection of the DNA that each of us contains in all of our cells. And I'll try and convince you of that. And one of my key points are simply that our genes make us what we are. So if we think about our genes or our genetic code as the blueprint for our bodies, then the result of this blueprint is, in the case of a blueprint, a beautiful home, and in the case of genes, ourselves. And I will have you know that I picked out these pictures very, very carefully. This one I found under Amy's dream house. And I hope you agree with me that I would look terrific in there and be extremely happy. But I am extremely happy anyways. And then I picked out this picture because you guys probably all recognize Rahm Emanuel. He has two brothers, Ari and Zeke. And I thought that these three brothers were an incredibly powerful example of the power of genetics, because each of them is extremely ambitious, extremely talented, extremely profane, and very, very successful in three very different areas. Ari is a Hollywood agent. Rahm, obviously, former chief of staff to the president and now mayor of Chicago. And their oldest brother, Ezekiel, is a very famous bioethicist and a professor at Harvard Medical School. Now, you might have known much of this. There was actually a really interesting article in the New York Times a few years ago called The Brothers Emmanuel. And in that article was an interesting story because these three brothers actually have a sister, an adopted sister. And she's had a very, very different trajectory in her life and has really struggled along the way. And I thought that was a really nice example of four individuals with the same environmental upbringing, but very different genetic codes, and how we are in many ways defined by our genetics. Okay, so as a real brief overview of the sort of terrain that we're going to cover, I'm going to do the first part of worms in men, and then Dr. Dubal is going to be talking about can we defeat brain diseases with genes that promote long life, and then Dr. Yokoyama will be presenting human neurogenetics, Homo sapiens as a model organism. Okay, so my part, I've organized into sort of three parts. The first is I'm going to give you sort of an outline or a rubric for how physicians and scientists can study neurodegenerative disease and actually other human diseases. Next, I'm going to tell you a little bit about what model organisms are and why they're useful. And I know Dr. Vossel covered this a little bit, but I'm going to take a little bit of a different approach from him. And then I'm going to give you one model organism vignette to try and illustrate the power of model organisms in understanding human diseases. Okay, so how do physicians and scientists study neurodegenerative disease? So this is a rubric, and this is actually a schema that I borrowed and adapted from a colleague of mine named Charlotte Sumner. And it goes in the following way. So an individual has to present to a clinician with a condition. And hopefully that astute condition will recognize the condition as something and then describe it. And once that condition is described, then additional persons with the condition can be identified. And as sort of a silly example, someone could come with green fingernails, and the clinician could say, that's not normal, and write a paper or a treatise on green fingernails, and then other people with green fingernails could be identified, okay? Now we have a cohort, or the ability to organize cohorts of people with green fingernails. And maybe, if you have that cohort, there's definitely more than one possibility. It could be that this is not a genetic disease, in which case the epidemiologist could go figure out, oh, there was something in the drinking water that gave people green fingernails. But if it is a genetic condition, or some subset of people with green fingernails have this because of a genetic change, then we can identify a gene or genes using techniques that Dr. Yokoyama is going to tell you about later on in the evening. Okay, once we have a gene or genes, we can start thinking about why that particular genetic change causes the green fingernails. Sometimes it's very obvious. Oh, the gene that makes nails green is overexpressed, okay? That is not true there's no gene that makes fingernails green. But there are, for example, tumor suppressor genes that are mutated. And as soon as we see there's a mutation in a tumor suppressor gene, we know exactly why that mutation causes the disease tumors. Usually it's not that simple. Usually it's a gene with a function that doesn't make sense or no function at all. And in that case, scientists need to work to determine what the mechanism of disease is related to that gene. And when I say gene, I'm actually talking about the product of the gene, which is a protein. Okay, so once you have a mechanism, you can develop targets for the therapy, hopefully, and then maybe you can develop treatments, and if you're really, really lucky, you could develop disease preventions, okay? So using this, I'm going to, this sort of scheme has been repeated over and over and over and over again in many, many different diseases. And these include neurological diseases such as epilepsy, migraine, and MS, multiple sclerosis, where single genetic mutations have been associated with these conditions. It could be cardiovascular or metabolic diseases like heart disease or diabetes, psychiatric conditions like depression, schizophrenia, mental retardation, and even a condition that we don't really consider a disease, I certainly don't, but is genetically regulated like aging. And Dr. Dubal is going to tell you a lot more about that later on in the evening. But because we're in the session called Genes in the Brain, what I'm going to give you an example of is how this scheme was used to study a neurodegenerative disease known as frontotemporal lobar degeneration, or FTLD. And I know you have heard about FTLD from, among others, Dr. Seeley and Dr. Miller, but I don't think any of them that I'm aware of use this rubric to describe how this disease we've come to understand it. So I'm going to do that. So this is a gentleman named Arnold Pick. And in 1892, Arnold Pick described a subtype of FTLD. At that time, it wasn't known as FTLD. He described a patient that had aphasia, or problems with language, and some behavioral and personality changes. And after he published his treatise, this condition became known as Pick's disease. Later on, he and others determined that Pick's disease preferentially affected the frontal and temporal lobes of the brain, so particular areas of the brain responsible for particular functions. And much later on, others determined that some cases of FTLD are associated with ALS, or Lou Gehrig's disease, which is a motor neuron disease. And based on that and other bits of information, we learned that up to 40% of these cases are actually genetically determined or familial. Okay, so now we've filled in the first 3 boxes here, all right? So a patient with frontal lobe symptoms presents to Arnold Pick. Pick's disease is described in 1892. And then once people have a syndrome, they can then identify more individuals with Pick's disease. All right, what comes next? Well, families. All right, let's say this person, a woman, because she's a circle, presents to a clinician with Pick's disease. And the clinician hopefully will do a careful family history and learn that, oh, her 2 sisters and brother also had the same syndrome, as did her mother and maternal grandmother. Now we have a big family cohort, and disease genes can then be identified. And using these types of large families, now we don't need as large of families, but back in the early days of human genetics, we needed families this big. Disease genes can be identified. And lots of disease genes have been identified, including tau or MAPT, progranulin, BCP, CHIMP2B, FUS, TDP43, and C9orf72. Now, that's just the list of genes. I'm actually not going to talk about most of these. I'm going to talk a little bit later about progranulin. But suffice it to say, we can now fill in the next 2 boxes. Families with Pick's disease and FTLD are organized, and then a bunch of genes are identified. Okay, so that, I made that sound really easy, but that actually took 100 years, over 100 years, okay? And probably millions of dollars and lots of sequencing. And that is pretty hard, but once you have these genes, there's still more work to be done. And how do these genes cause this syndrome? We want to understand that. So how do we do that? Well, we can't really do experiments on people. And so we turn to our friends model organisms. All right. So what are model organisms and why are they useful? So here we are, the tree of life, and Homo sapiens is perched here right at the top. And from our lofty perch, we can view all of our friendly model organisms. And this would include things like Saccharomyces cerevisiae, or the baker's yeast, which is probably the single most important single cellular model organism that we have. We've learned so much from yeast. It would be an exhaustive lecture to tell you about what we've learned from yeast. But one example that stands out in my mind is from yeast, we've learned basically how proteins are trafficked around the cells. And loads of things can go wrong when proteins are not trafficked correctly. Okay, so that's one of many things we've learned from baker's yeast. Here's from our arthropods, we have our friendly fruit fly, Drosophila melanogaster. Now, I shouldn't say this, but I am not overly fond of fruit flies. And it's because I was in a lab where there were fruit flies right next door, and they were constantly buzzing around. You'd pull out a banana, and then zoom, here they come. But that being said, we have learned a lot from fruit flies as well. And one example I like to give is that from fruit flies, we've learned a lot about patterning. And by that I mean how axially symmetrical organisms, like ourselves, become so. So let's say there's 2 cells. One cell needs to go right, the other cell needs to go left. How do those 2 cells know which direction to go? Well, that's a genetically regulated process. So we can make genetic changes so that both cells go left, or both cells go right, or the cells sort of wander off where they're not supposed to go. And we've learned a lot about this type of directional patterning from Drosophila as well as many other model organisms. My personal favorite is C. elegans, or the roundworm, Cenorhabditis elegans. And I'm going to tell you a lot more about C. elegans in a little bit. But before I do that, I'm going to go over why we use model organisms, okay? So what I've already mentioned before is that obviously there are ethical considerations in human research. We just can't be doing experiments on our neighbors or our neighbors' kids or our neighbors' parents, okay? In many cases, the basic biological processes involved are conserved in humans and in model organisms. A cell is a cell. Most cells have a nucleus. They have ER. They have a Golgi. They have mitochondria. And so we can study these processes in model organisms as well as we can study them in humans. Probably better. Also, model organisms allow us to control genetics and the environment around the genetics. So we can control temperature, we can control light-dark cycles, we can control diet. We can also amplify and repeat studies. I can grow 1 million C. elegans in the lab without blinking an eye and without breaking a sweat, and that's pretty powerful. And there are loads of tools and techniques available, molecular, genetic, biochemical, that we can use. And finally, we use them because they work. We know they do. I've told you a bunch of things that we learned from model organisms, and in a few minutes I'm going to tell you some more. Okay, but now on to my favorite, C. elegans. So C. elegans, like the other model organisms I've told you about, oh, and I've left out mice, but Dina is going to tell you more about mice. That was an oversight on my part. And that makes me think of them because mice actually have spines, but the creatures I talked about are tiny, spineless, and brainless. So what in the world can they teach us about neurodegenerative disease? Well, C. elegans is called C. elegans because it has an elegant tapered shape. And it looks giant and scary here on the screen. But a fully grown adult C. elegans is about 1 millimeter long. So it's actually a microscopic worm. And it's now called a rotting fruit nematode. I showed a picture of a compost heap because that's where you can find them. Anyone with a compost heap in this room can go out and dig out some of the dirt at the bottom, and there will be tons of C. elegans in your compost heap. C. elegans are wonderful for many reasons. They are small and easy to grow. Like I said, I could grow 1 million of them in the lab just on Petri dishes. They have a very simple anatomy. They have a mouth here that's connected to a pharynx with a grinder to digest the bacteria they eat. They have an intestinal system. They have, actually the majority of their body is taken up by their reproductive system, and they have neurons all spread throughout. They're genetically tractable, meaning we can make genetic changes in them. We can knock down genes, we can introduce new genes, and we can keep what are known as isogenic populations because C. elegans are hermaphrodites. They produce both eggs and sperm. They're either hermaphrodites or they're males, so they're either XX or XO. And they're transparent, so we can look at them under the microscope in real time. And they have a lot of complex behaviors, more so than one might think. So we can measure their movement, we can measure their reproductive abilities, we can measure actually their ability to learn and to forget. Okay, so if you're still not convinced, I now have a list of things that we've learned about from C. elegans. All right, so one of them is this concept, this whole idea of programmed cell death. So programmed cell death is the normal death that occurs during development and sometimes in response to injury or if we have cells that we don't need. And the initial molecules that control programmed cell death were first identified in C. elegans, and that's because the entire lineage of C. elegans of every cell was described, and so scientists could watch cells be born and die. You may or may not have heard of RNA interference. This is a concept whereby DNA makes RNA and RNA makes protein. And we used to think RNA was just this intermediate between DNA and proteins. Turns out RNAs do a lot more than that, and there's a lot more to be discovered about what RNA does. Well, one of the things RNA does is that certain little RNAs can control the production or the activation of other RNAs. And so this concept of RNA interference was actually born in plants, but sort of brought to fruition in C. elegans. There's something called green fluorescence protein that I think Dr. Vossel talked about. It's this glowing protein that was isolated from a jellyfish. But because C. elegans are transparent, the initial work done with GFP, or green fluorescent protein, was done in C. elegans. Dr. Dubal is going to tell you a lot more about this, but the whole idea that aging is not just things falling apart, that aging is actually genetically regulated, was discovered in C. elegans, in part in the lab of Dr. Cynthia Kenyon, who's in this building upstairs in Genentech Hall here at UCSF. And because we were talking about neurodegenerative diseases, there is a gene known as presenilin that is known to be associated with familial forms of Alzheimer's disease. And that gene was first described in C. elegans as well. And I bring up these 5 examples because I like to put little copper pennies next to them. And these are actually not copper pennies, they're Nobel Prizes. So in the last 12 years, 3 Nobel Prizes have been awarded to scientists studying primarily C. elegans for work in programmed cell death, RNA interference, and the use of green fluorescent protein, all of which have revolutionized our understanding of human health and disease. Okay, so that's great, but does C. elegans have a brain? So not officially, and certainly not as beautiful and complex as a human brain. But it does have neurons, and that's important. And the other nice thing is that we know exactly how many neurons C. elegans have. It has exactly 302 neurons. And not only that, we know every single connection that every neuron makes in C. elegans to other neurons. We also know for every neuron what neurotransmitters it makes. So I think you guys heard a little bit about, or maybe you didn't, maybe I was reading that. There are chemicals that neurons use to talk to other neurons. And the same neurotransmitters are used by C. elegans as we use in our brains. And as I mentioned, there are many behaviors that we can study in C. elegans, including coordination, reproduction, chemotaxis, learning, and memory. Okay, so I'm going to finish up by telling you about one model organism vignette. And that has to do with the protein progranulin. So I mentioned to you that progranulin in humans is mutated in some forms of frontotemporal lobar degeneration. And that discovery was actually made recently in 2007, or actually late 2006 that those papers were published. And lo and behold, C. elegans has also a progranulin protein. And this is a model of the human protein, and it has a signal here that tells the cell that the protein progranulin should be secreted out of the cell. And the same signal is here in C. elegans progranulin. And then these red boxes are individual granulin domains, which is just a protein motif that is also conserved in C. elegans progranulin. As is often the case, C. elegans progranulin is a little bit simpler. It has fewer granulin domains compared to human progranulin. But more isn't necessarily, there's no value judgment necessarily on more in terms of is it better or is it worse, because as it turns out, zebrafish have like 10 granulins, whereas humans have only 7.5. Okay, so when it comes to progranulin, balance is essential. And by that, I mean that too little progranulin is not good. That's what happens in neurodegenerative disease. There's not enough progranulin produced. But too much progranulin is not good either, because very aggressive tumors that grow fast and metastasize early tend to produce lots of progranulin. Okay. So what does progranulin do? Well, based on what I just told you, too little progranulin causes neuronal death. Too much progranulin causes tumors to grow. One could posit that progranulin is something like a growth factor. You don't have enough of a growth factor, things die. You have too much of a growth factor, tumors go crazy. So that was sort of how people thought about progranulin. Initially, they thought, okay, you have progranulin, it's a growth factor and it improves cell growth and survival. So the model would be that if you don't have enough progranulin, as in neurodegenerative disease, then you get decreased cell growth and survival, and then you get neurodegeneration. And that is absolutely a very attractive and a very appropriate model, except for it totally was not true. Okay? People tried to knock down progranulin and look to see what happened to neurons. They were fine. So what does that leave us with? So it was around this time that we thought, well, worms have a progranulin, C. elegans has a progranulin. Let's see what happens to C. elegans that don't have progranulin. So how do you get a C. elegans that doesn't have progranulin? Well, as it turns out, there are two groups in the world, and what they do are they just mutagenize worms and figure out what genes are not normal in them, okay? So what do I mean by mutagenize? So we take this worm, and there are different ways to do it, but the most common is to just treat the animals with a chemical called EMS that causes DNA to break open, and then it's repaired sometimes correctly and sometimes incorrectly, and you get mutations in genes. And then you get loads and loads of genes or you get loads and loads of animals with different gene mutations, and then with sequencing techniques and other techniques, lo and behold, we found a worm that is missing progranulin. And when I say we, I don't mean we. Actually, it's these knockout consortia. All I did was put in a request on the computer, and 2 weeks later, someone shipped me a plate of C. elegans that were all progranulin mutants, okay? So we got those worms, and we started to characterize them. And the first thing we noticed is that worms that don't have any progranulin actually look pretty normal. And that's okay. We did some initial characterization, and their lifespan is normal. And we looked at their reproduction, and their reproduction is pretty normal. It's a little bit decreased, but overall they looked fine. So we realized that we were looking for more of a subtle change or a subtle phenotype in these worms. And it still left us with the question of how does insufficient progranulin cause neurodegenerative disease? Okay, that's our big question. And the answer to that actually lay partially, probably, hopefully, lies in the concept of programmed cell death. Okay, so let me tell you a little bit more about programmed cell death. So programmed cell death is a very fundamental biological process. This is an egg that hatches and turns into a tadpole, and eventually that tadpole, as we all know, turns into a frog. Well, there's a big change that happens here, and one of the things I like to point to is the fact that this frog has a tail. So how does that frog get rid of that tail and become a mature frog? That tail, the cells in the tail have to, we would say, shrivel up and fall off, okay? But if we were to use more precise terminology, what is actually happening to the cells in that tail is that they are all undergoing programmed cell death. So programmed cell death happens to cells when they are no longer needed, when they're damaged, or when they're extraneous, okay? And so as it turns out, this process of programmed cell death is very complex, but it's also conserved, okay? And it's not important to see all the genes in here. But a very similar process of programmed cell death helps the webs that are between our fingers go away as we develop as embryos, okay? So a very, very similar process. Okay, so I've told you that we learned a lot about programmed cell death from C. elegans. And the way we did that is because this guy, John Solston, went through and carefully cataloged. He literally sat in front of a microscope for probably thousands and thousands of hours from the single egg stage of a C. elegans through, 16 cells, 64 cells, and he watched every single cell divide, and he cataloged exactly where they were. And then he published this paper, which showed basically the division and lineage of every single cell. And this is just one page of 13 pages of C. elegans lineage. And I'm circling a couple of these little things. They're little X's, and those are cells that die, okay? So once we knew the exact fate of every cell, then scientists, both here in the US and in Europe, developed techniques to automate how the tracking of these cells. And based on that, we could start to measure the life and death of cells much more carefully on a minute-to-minute basis. And we were able to learn about things like the time to die. So that would be the time from when a cell is born. Let's say this cell is born. And then, let's say this cell is born. And then if it's fated to die, how many minutes it's alive before it starts to die. And once the cell starts to die, and dying cells take on a particular appearance that a microscope we could recognize, but also an automated microscope can recognize. And then we can start to measure things like time to clearance, how long it takes for a cell, once it starts to die, to be completely eliminated. And what we learned is that progranulin mutant C. elegans have a problem with their time to clearance. So in other words, worms lacking progranulin exhibit abnormal programmed cell death. So this is once again an embryo, and it shows 3 cells that are in the process of dying. And if this were a cell that I were watching under the microscope, I could literally watch those cells die. And what happens normally is that a cell goes through, a dying cell goes through a particular maturation process, and then it disappears because it's been engulfed and eaten up and digested by its neighboring cell. And that process takes, on average, 10 minutes, normally. But if a cell doesn't have enough progranulin, then the same process of maturation occurs, but it takes about half the time. Okay. So what that says is that progranulin normally slows the clearance of dying cells. So here we have a dying cell, and there's a partner in all of this, the engulfing cell. And if there's not enough progranulin, this dying cell takes about half the time to disappear. And we weren't sure if it was because of this cell doing something more quickly, or if it was because of this cell recognizing and engulfing the dying cell more quickly. And in order to figure that out, we looked at engulfing cells, or macrophages, from some worms that some colleagues of ours over at the Gladstone Institute across the street had made. And these were, not worms, they were mice. And we were able to look at mice that normally had normal levels of progranulin, and if we expose these cells, which are these sort of gray shadows, to fluorescent beads, they will eat the beads. And if there are normal levels of progranulin in these macrophages, they'll eat a certain number of beads. But if there's no progranulin, they eat a lot more beads. And so what that tells us is that progranulin actually normally slows the process of engulfment, or the ability of phagocytic cells or engulfing cells to recognize and eat beads. And in experiments I'm not going to show you, it also does the same thing for dying cells. So macrophages without any progranulin are able to recognize and engulf dying cells better, faster than wild-type macrophages that have normal levels of progranulin. Okay. So I've told you when it comes to progranulin, balance is essential. If you don't have enough progranulin, you get neurodegeneration. If you have too much progranulin, tumors grow fast and metastasize early. And we've now implicated this process of programmed cell death in this imbalance. Okay? So what we've basically shown is that progranulin is a signal that regulates the rates of engulfment. And basically, we love that because it immediately presents a model for how changing progranulin levels can cause neurodegeneration and tumors, and that is the following. If you have normal levels of progranulin, let's say we have a healthy cell, and let's say that cell gets injured, and I didn't tell you this, but an injured cell starts to act a little bit like a dying cell, this spectrum, okay? But let's say, by definition, our damage or our injury is non-lethal. So that injured cell starts to look a little bit unhealthy. But because there's plenty of progranulin around, the neighboring engulfing cells know, oh, I'm not going to recognize it and eat that damaged cell. And so now the cell can recover, and as opposed to being recognized and removed and meeting a premature death. So let's say there's too little progranulin. Now you have an injured cell, and there's not enough progranulin to sort of slow down those aggressive macrophages, those aggressive engulfing cells. And so now the cell could be engulfed prematurely. And you could imagine over a lifetime, too little progranulin could lead to a condition like neurodegeneration. However, if there's too much progranulin, that's not good, because tumor cells could use secrete loads and loads of progranulin to basically shut down the cells that should be recognizing and engulfing them. And then a tumor can then evade these surveillance cells as opposed to being recognized and removed like they should be. Okay, so now if we go back, we've made our way around to here, and now we've shown that progranulin deficiency can accelerate the engulfment of dying and maybe injured cells. And it gives us a new target for therapy, and that is programmed cell death. Okay. Now, I would love to be able to stand up here and tell you we've done this next step. We've developed the treatments. But, this gene was recognized as being a disease gene just 5-6 years ago. And so this is we're still working on this step. And hopefully, in a little bit, we'll be able to come back and tell you, yes, we've succeeded in this. But, every step along the way has sort of trials and tribulations. And so maybe I can report back. But I don't want to be too pessimistic. And so I'll tell you a little bit of some promising work that's been done by another group. Okay. So the whole idea is in neurodegeneration, you have not enough progranulin. And that's because in the disease, there is one normal progranulin gene, and then there's a progranulin gene here with a mutation. And from this abnormal gene, there's no progranulin protein produced. So if we want to restore the balance of progranulin, it'd be nice if we could get some progranulin produced from this progranulin gene. And so some of our colleagues in this consortia to study FTLD, that's known as the Bluefield Project, have been trying to find ways to increase the production of progranulin. And this is work from Joachim Hertz and Gang Yu's labs at UT Southwestern. So they took FDA-approved compounds, so a library of chemicals that are already approved for other uses, and then they just took the chemicals and they put them on cultured cells and measured the amount of progranulin those cells produced. And they found a compound called compound 1 that increased the amount of progranulin that these cells could produce. And actually they found another one too that also increased it. And then they did this neat trick with another wonderful model organism, mice, where they attached something that glows to the progranulin, and then when they gave the compounds to the mice, they could actually just see how brightly the mice glowed. And if they glow really brightly, it means they're producing lots of progranulin. And so there is definitely bright spots on the horizon. Okay. And I didn't even mean to pun, but I did. Okay, so that's basically my section. So to summarize, what we've discussed are that the study of human diseases really relies on the contributions from multiple disciplines: clinicians, epidemiologists, human geneticists, scientists that study model organisms, multiple disciplines. Model organisms such as yeast, worms, and flies have been and continue to be very important in understanding human diseases and developing therapies. And I know you've seen a picture of this beautiful jellyfish before, Eichoria victoria, but I always like to put in a little plug for the fact that the long-term benefits of research endeavors really can't be predicted. When people started studying jellyfish, they didn't know that they were going to revolutionize biology by discovering green fluorescence protein. And so basic science research with lots of model organisms really needs to continue to be supported. And with that, I'm going to turn the floor over to Dr. Dubal.
Well, good evening. I'm Dina Dubal, and I'll begin with an introduction. I have a similar path to Dr. Kao here. I am a physician scientist. So I'm of Indian origin, born and raised in the U.S., but actually raised in the South, so I used to have a Southern accent. Which left me when I went to UC Berkeley, where I studied anthropology and molecular and cell biology with an emphasis in neuroscience. And it was at UC Berkeley where I became just completely fascinated with aging. And I worked with an anthropologist, Dr. Lawrence Cohen, a medical anthropologist, to begin to think about and understand how did people experience aging and disease differently across cultures. How is it different to have dementia in India versus the US versus England? What sort of social influences, economical influences, cultural influences? How did the disease manifest differently cross-culturally? And then in my neuroscience classes, I became very aware of how much we didn't know about these, about aging or diseases of the aging brain. And so, so between my anthropology and neuroscience background, I pursued an MD PhD at the University of Kentucky at the Sanders Brown Center on Aging and Department of Physiology, and really focused my study on aging processes there, and with the idea that I really wanted to learn about the aging process and about disease enough to really help in the development of treatments of these diseases that affect people across the world in every culture. So from Kentucky, after getting my medical and PhD training, I came to UCSF and have been here for almost a decade, where I became a neurologist and then a specialist in aging and dementias, with just incredible mentors and an incredible training process here at UCSF. So today I would like to share with you my passion for aging, and also to share with you what is on the horizon, and how can we think about aging, genes, brain, and disease. So the title of the talk here is, Can We Defeat Brain Disease with Genes That Promote Long Life? And the short talk is yes. But I actually do have a talk. So the world's population just exceeded 7 billion people, and it is aging rapidly. And the portion of the population that is actually aging most rapidly is the age group 60 and older, and this is occurring across the world. And this really poses a huge challenge for us, because we must ask ourselves, how will we achieve healthy aging? So it's absolutely critical from a scientific standpoint to really understand the aging process, to then know how to achieve healthy aging. So I just showed you a picture, actually, of what the economists portrayed as an impending disaster here. The world's population is aging, and it's gloom and doom. We don't have the economics to support the cost of aging individuals. We don't have yet the scientific discoveries. I mean, this looks like a disaster. But with interventions, this will be a celebration. So let's actually step back and celebrate for a moment, and look at what's happened over the past century or so. So what you see here is a graph of lifespan, and what this shows you is that in the 1800s, in 1850, people were living, and this is in developed nations, USA, Japan, in Europe, people were living to be approximately 45, 50 years old. And look at what's happened over a relatively short period of time. In developed nations, we are now living to be into our 80s. This is remarkable. We've almost doubled the lifespan. So we now have the ability to age, and this accomplishment marks a huge improvement in global health. We have conquered many, many problems that are still being conquered in developing nations, but infections, sanitation, we can even have a heart attack, and we have the technology to survive and live a normal life after that heart attack. But now what we would all argue here, our biggest challenge is the brain, and how do we preserve our aging brain? We have no therapies, and in fact, without interventions, the economist's portrayal of this impending explosion will be true. But there are things on the horizon, and we are optimistic. But what we do face dramatic increases in chronic diseases of the aging brain, such as Alzheimer's disease. So Alzheimer's disease is the leading cause of dementia, and many of you know from relatives or friends. We treat patients here with Alzheimer's disease. The symptoms really begin insidiously. They begin slowly with mild memory impairments that then spread across the brain to involve deterioration of judgment, of language, orientation, and ultimately cause a bedbound condition. This is a devastating neurodegenerative disease, one of many. And the development of therapies to cure Alzheimer's disease is urgent. By the year 2040, over 80 million people will have Alzheimer's disease in the world without interventions. So let's look further into a brain. I'm going to show you a cartoon and then talk a little bit about how we model Alzheimer's disease. So interestingly, a small percentage of Alzheimer's disease is caused by genetic mutations. And we take advantage of this by introducing those genetic mutations into mice. So what you see here actually is a cartoon of two neurons. And these red dots here are toxins. They're amyloid beta. You've heard about them earlier in the course. All genetic causes of Alzheimer's disease are caused by mutations in the human amyloid precursor protein that causes A beta. Every single mutation affects A beta. And so A beta becomes an important toxin to understand. So here's a cartoon of what A beta does, in that it assembles itself into these toxic, we called oligomers, and it attacks processes of brain cells, it attacks the brain cells themselves, and it essentially wreaks havoc on brain cells, individual cells, the circuit of cells, brain-wide, and essentially affects memory and the other cognitive functions that we talked about. And so what we do is we study Alzheimer's disease using genetic manipulations in mice. And so here we go. This is a human brain that's affected by Alzheimer's disease, and it is it's shrunken, it's degenerated. Now, this human brain suffered from a specific genetic mutation of the human amyloid precursor protein. The model that we use is introducing that genetic mutation into the mouse brain. And so essentially we've produced what some people call Mouseheimers, but mice with a touch of Alzheimer's disease. And what you see in the human brain in terms of amyloid plaques, you've heard that earlier in the course here in red, and dystrophic neurites, we see the same thing in the mouse models of Alzheimer's disease. Most importantly, what we see is that the mice that model Alzheimer's disease show very similar cognitive, learning, and memory problems that humans that suffer from Alzheimer's disease show. And so we do not have a perfect mouse model of Alzheimer's disease, but we have very good strong models of Alzheimer's disease, and I'll be showing you some data from such a mouse model. Back to aging. How can we connect aging with Alzheimer's disease? So it's actually quite easy. Aging is the biggest risk factor for Alzheimer's disease. It is the primary risk factor for Alzheimer's disease, and with every 5 years, 5 years after the age of 65, the risk doubles for Alzheimer's disease. And so with increasing age, there's an increasing risk for Alzheimer's disease. Now, Dr. Kao alluded to one of the most, I think, remarkable discoveries of the past decade, and that is that the aging process is a plastic process. And this was primarily discovered in worms in an effort that was really spearheaded by Cynthia Kenyon's lab where Dr. Kao trained. So the aging is a plastic process. What does that mean? It means that actually tinkering with specific genes, with just 1 or 2 molecules, can have dramatic effects on how long an organism can live or how short an organism can live. Tinkering with very specific genes or molecules have dramatic effects on lifespan. And so that aging itself is amenable to change. So this raises an intriguing and high impact question, and that is, can regulators of aging prevent degenerative disease? So if one can slow down the aging process, can one prevent degenerative disease? In other words, instead of perhaps targeting A beta or targeting tau, these toxins that you've heard about before, what about targeting the biggest risk factor for developing those diseases, aging? So this is the viewpoint that my lab focuses on, is really looking at intersections between aging and dementia, looking at anti-aging factors to see if they are effective agents in preventing diseases of the aging brain. And today I will talk to you actually about one focus of my lab, which is female longevity. And so the premise here is this. The premise is, if we can understand basic fundamental mechanisms of aging, of increasing lifespan, can we harvest those mechanisms, apply them to men and women to prevent diseases of aging? That's the premise. And the idea is not to live to 300 years, and worms can actually live 3 to 4 times longer than normal, and so that would be equivalent to us living 300 to 400 years. The goal isn't to live to 400 years. The goal is to live a very healthy, long life. And what worms and mice have taught us is this, that in fact, these anti-aging factors confer health as well, not just long life. So I'd like to talk to you about female longevity, which is a fundamental biologic phenomenon that my lab studies and is actually not understood. We know anecdotally that women live longer than men, but they really do live longer than men across the world. And so this is data taken from the World Health Organization last year, showing you that in both developed, like the US and Japan, and developing nations like Uganda, women are living longer than men by 5, 7, even more years. This is true across the world. It's true across socioeconomic status. And there certainly may be cultural and environmental factors, but I would argue that there is something even more than that, because female bears live longer than male bears. Female whales live longer than male whales. And this effect of female longevity is conserved across the animal kingdom. And so there is something fundamentally biologic about female longevity. that has yet to be understood. And in fact, if one were to take a brain cell from a female mouse, put it in a dish, so that's an XX cell, it's a female chromosome, it would be an XX carrying neuron, and then compare it to an XY, or a male neuron from a mouse brain, the female brain cell actually lives longer than the male brain cell. Remarkable, even in a dish. So what we're studying is what is that? Is that something that is genetic? We think so. And can we, again, harvest that mechanism, understand it, and turn it into a therapy for men and women? Augment it and turn it into a therapy for men and women. An unconventional approach to thinking about treating neurodegenerative disease. Now, this phenomenon of female longevity also applies to neurodegenerative diseases. And so let's talk about Alzheimer's for a moment. More women have Alzheimer's disease. They live to ages where Alzheimer's disease is more prevalent. They live longer. What about the risk of developing Alzheimer's disease? In some populations, it's more in women. In some populations, it's more in men. All in all, across the world, studies have shown that it's pretty similar. The risk of developing Alzheimer's disease is pretty similar between men and women. An aspect of the epidemiology, meaning just the human study of Alzheimer's disease that has received very little attention, is the course of the disease. And what has been shown, and what one observes in group after group after group, is that males with Alzheimer's disease progress to death faster than females, suggesting a more aggressive course of the disease compared to females. This has been reported in many in many papers, where you can see that in red, the women with Alzheimer's disease will do die, but they die at a slower rate compared to men who have Alzheimer's disease, who progress to death faster, suggesting that perhaps that aspect of female longevity, which one sees in normal aging and in neurodegenerative disease, maybe there's something protective there that we can understand and then harvest again into a therapy to treat both sexes. Well, this has been shown it's been shown in multiple populations, but we thought, let's look at this ourselves in our own population here at the Memory and Aging Center. This is several thousand patients, and we see the same thing, actually, that men with Alzheimer's disease progress to death faster compared to women with Alzheimer's disease, here in red. Okay, well, let's model the disease and learn something and identify therapeutic targets to defeat the disease. This is only possible if our animal models of disease recapitulate the human condition. So an animal model is useless if it's not really modeling the human condition well. So then we looked at our Mouseheimers, and we found the same exact thing. So this is thousands of mice that carry that Alzheimer's genetic mutation, and we found strikingly the same thing. That is, male mice with that human amyloid precursor protein that causes Alzheimer's-like problems will progress to death faster compared to the females. And so it leads us to the hypothesis that there might be neuroprotective targets that are related to sex, and maybe there's something here that we can understand, harvest, and apply to both male and females. But there's a fundamental difference between mouse aging and human aging. And it has to do with aging of the reproductive system. In humans, gonadal steroids, estrogens, testosterones, progesterones, are decreased with aging. And this is true in both males and in females. And so in the post-reproductive years, gonadal steroids will decrease dramatically in humans. If one were to look at total testosterone in males, it would be a little higher, but this is biologically active testosterone. This is biologically active estrogen and progesterone. Gonadal steroids decrease in human aging. In mouse aging, they don't. Now, mice undergo reproductive aging, which means that that sperm quality decreases, that females go through their own mouse menopause, but they don't lose their hormones in the way that humans lose hormones. And so in a young mouse compared to an aged mouse, hormone levels are essentially, they remain high. So in our studies, we recapitulate the human condition. When we use our mouse models of Alzheimer's disease, we take away the hormones to model what happens in the human condition. Okay? So what we did is we removed gonadal steroids from male and female mice to mimic what happens in human aging. And then we asked, okay, with hormones aside, what is it that is impacting female longevity? Is it effects of hormones that last even beyond their presence, an organizational effect of hormones? Or could it be something about sex chromosomes? What underlies female longevity? Now, to attack this question, we used a really unconventional genetic approach. And so this is going to be a little bit of a puzzle and maybe a brain teaser. So it'll require some focus here. What we did is we dissected in a genetic way whether sex chromosomes or long-lasting effects of gonadal hormones affect female longevity. Okay? And the way we did this was the following. We took an XX mouse with normal autosomes. So we have all mammals have one pair of sex chromosomes and then a bunch of other chromosomes that are not sex chromosomes. They're called autosomes. Those are where our genes are located. And so XX is a sex chromosome in females. XY is a sex chromosome in males. And A represents autosomes, all the other chromosomes. Okay, so what we did is this. We produced mice that have female sex chromosomes, but male gonads, and vice versa. Male sex chromosomes and female gonads, and vice versa. And I'll take a moment to explain this because it's, again, a really fascinating scientific experiment that is going to tell us something very, very interesting and fundamental to biology. So this is how the mice are made. We cross, which this is what that line means, an XX, a normal female mouse, with a male mouse that has a gene that's taken off of the Y chromosome and put onto an autosome, and that gene is called SRY. It's the testes determining factor. That gene makes a protein that causes male gonads to form. And so with the presence of that gene, SRY, the testes determining factor, testicles are formed and male differentiation occurs. Okay? So imagine that if this is taken off of the Y chromosome and put on another chromosome, if you mate these two, you can get 4 sex phenotypes now. You can get XX mice, just normal XX, without that testes determining factor that produce ovaries. You can also have XY mice without that SRY, and without that SRY, there are no testes produced, ovaries are produced instead in a default manner. Although the biology is more complex than simply default, but they produce ovaries. These male XY mice that have ovaries are phenotypically female. That means they look like females, they act like females, they have cycles, they have female reproductive behaviors, they can't get pregnant, but for all intents and purposes, they're female. It's just that their sex chromosomes are XY. And then one can get XX female mice, but they inherited that SRY on their other chromosomes, so they produce testes. These XX female mice are male. You cannot distinguish them from other males. They have testes, they produce normal levels of testosterone, they show aggressive behaviors, they mount females, they ejaculate, they're male, but they're XX. And then the fourth sex phenotype here is the XY without that SRY, but then the SRY is on the other chromosome, and so they do produce testes and are phenotypically male. Okay, so this is the unique genetic manipulation here, but what we get at the end of the day are 4 sex phenotypes. And now, brain teaser. This is how we dissect the effect of either gonadal steroids or sex chromosomes. If these mice that are circled live longer than these mice that are underlined, in other words, if the mice that have ovaries live longer than the mice that grew up with testes, that would tell us that female longevity was dictated by effects of gonadal steroids. Okay? If mice here in red that were XX, regardless of having ovaries or testes, lived longer than mice that were XY, didn't matter whether there were ovaries or testes, that would tell you that sex chromosomes dictate female longevity, okay? And so here comes the decisive experiment. So which one is it? Either one would be equally exciting and important and lead us to understand a fundamental mechanism of aging. Is it sex chromosomes? In fact, it is. And this was a really recent and remarkable discovery that I'll outline here. So what you see here are survival curves of the mice, of the Alzheimer's mice. And what you can see is that XY male Alzheimer's mice in blue progress to death faster than the XX Alzheimer's mice in red. We know that. We see that in the human population. We saw that in that big mouse population I showed you. But now we switched out their chromosomes or gonads, and we see that in orange here, XX, even if they grew up with testes, they live longer than the XY who grew up with ovaries. Okay, so what this experiment shows is that XX mice, and this is the Alzheimer's model, that XX mice, regardless of whether they grew up male or female, live longer than the XY. So this means that there is a sex chromosomal basis for modulating, impacting longevity. So now what we've done is we've really uncovered a new field that we are now figuring out. Is this because of the Y, genes on the Y, genes on the X? What is it? We know it's sex chromosomes that impact longevity. What is it on the sex chromosomes? That's ripe for discovery, and can those be therapeutic targets for men and women? So lifespan, very clear results. What about cognition? What about the learning and memory? We saw the same thing. You've heard several weeks ago about the Morris water maze in mice, and that is a test of learning and memory, where mice map their spatial environment to find a hidden platform in cold water. And those that travel a quicker distance to find that platform over a smaller period of time learn faster. So this is water maze data that has a lot of lines, but let me simplify it. If a mouse learns fast to find that hidden platform, the line goes from here down to here. If it's not learning fast, it's traveling sort of the same distance over several days. It's just not learning to find that hidden platform. So the bottom line of these graphs is that the mice that were X with Alzheimer's, didn't matter whether they were female or male. They were impaired. They have Alzheimer's, but they did eventually learn. They had a less severe impairment compared to the XY mice, whether they were male or female. Didn't matter. If they were XY, they had a more severe disease. So they had a hard time finding that hidden platform, traveling, 1,000 centimeters. It's just not improving over time, showing a more severe cognitive deficit. So what these data show is that XX mice that model Alzheimer will learn better in the water maze. They're not completely protected. They still have cognitive impairments, but they're less severe cognitive impairments compared to the XY mice with that model Alzheimer's. So, sex chromosomes impact longevity and learning and memory in aging and disease. And we are identifying again, what are those target genes on those sex chromosomes? 3 take-home points. Aging is a plastic or a changeable process. If we understand fundamental mechanisms of aging, this is a really promising route for therapeutic targets. Also, understanding the fundamental mechanisms of aging, such as female longevity, will provide treatments against diseases of aging for both men and women. There's something to be learned from this natural phenomenon of women living longer, of females living longer. And our genes, including those on our sex chromosomes, robustly influence longevity and resistance to brain disease. And these are the acknowledgments. So I'm a new lab, the Dubal lab here at UCSF, and in the new Sandler Neuroscience Center. And we're building our lab, and we are extremely dedicated to these questions and really hope to have some novel therapeutic targets. These are my mentors, Leonard Mucke and Bruce Miller, and multiple collaborators, both at UCSF and at other institutions. If you are ever interested in visiting the lab or seeing the new building, please contact me or any of us. I think that's an open invitation. We would love to show you around and share how the science works. So thank you so much.
So I have to apologize in advance. I came down with a full-fledged cold yesterday, so hopefully you'll be able to hear me throughout, but if I turn my head to clear my throat, hopefully you'll pardon me and you can take a little stretch in the meantime. So my name is Jennifer Yokoyama. I'm a postdoctoral fellow at the Memory and Aging Center here at UCSF. And so I have a PhD in basic genetics research. And actually, I got my PhD at UCSF as well. But in my former life, I was actually a canine geneticist. So I was in the lab of Steve Hamilton in the Department of Psychiatry and Institute for Human Genetics. And we were using the domestic dog as a genetic model for studying anxiety disorders. And so there's actually these anxiety disorders that occur in certain breeds of dogs at a very high frequency. And there's no genetic manipulation. It just naturally occurs. But I just wanted to let you know that because as part of this talk, although I'm working on humans now, I have a very fond place in my heart for model organisms. And so today I'm going to tell you about how we use humans in neurogenetics research. But I want to hopefully, at the end of this talk, kind of wrap it back around so we can think about how human genetics kind of fits into this context of using model organisms in a very complementary way. And so, since the late 1800s, when Gregor Mendel first proposed these theories of heredity via his studies in pea plants, the world of genetics has really come to this place where we're very technology-driven and fast-paced. And just to give you an example, in 2001, the human genome was published, and this genome had taken many, many years to sequence, millions and millions of dollars to fund, and yet, here we are, just 10 years later, pretty much, and we're on the brink of being able to sequence an entire human genome for about $1,000 and in about a day. And so, since in 1990, when the Human Genome Project was first started, we've if the red blob kind of represents the types of research that we're doing using genomics, we've moved from this era of just trying to understand structures and biology of genomes to really into this kind of present day where we're starting to understand biology of disease. And hopefully within the next 10 to 20 years, we can really start making really big advances in science of medicine, as well as making big impacts on the way we deal with healthcare and genomics. And so I've divided my talk into kind of three little parts. The first part, I'm going to give you a basic introduction into the world of human genetics. Next, I'm going to tell you about 3 different types of genetic studies that we commonly use in this field. And I'll give you little examples of some recently published research for each of those types of studies. And then I'll wrap up to just talk a little bit about where we think the world of neurogenetics is going in this kind of technology-driven era. And so perhaps after this talk, you'll be able to go home and open up your human genetics journal and read all the papers and understand all of them. But if that's not the case, minimally, I hope you can go home feeling like everyone can be really excited about genetics. Because as you can see, the 3 of us are all very excited about genetics, and we think it's a really great field. So as Dr. Kao mentioned, we have these very great model organisms that we can use to study genes in really detailed ways. But for ethical reasons, we can't really do the same thing in people. And so instead, we rely on, cute costumes and naturally occurring genetic variation in the human genome. And so we can utilize differences between groups for gene discovery. And for a lot of the talks that you've heard in the rest of this seminar and the things I'm going to be focusing on today will be really related to discovering genes related to neurodegeneration. So why are genes important? Well, by identifying genes that are associated with diseases, we can start elucidating some of the underlying biology that describes that disease. And in addition, we might be able to implicate some new targets for therapeutic interventions for treating those diseases. In addition, as you probably know, at the moment of conception, our genomes are basically present, and we carry those, albeit somewhat modified in different ways, basically for the rest of our lives. And so we have the promise of the genome as a very early marker for trying to make predictions on disease risk. So just to give you one example of how gene discovery has really aided in the understanding of a disease, in the early 90s, ApoE4 was discovered as one of the main risk factors for Alzheimer's disease. And in the last 20 years, researchers like Yidong Huang here at the Gladstone Institute have been able to really start trying to tease apart all of the molecular mechanisms for how the ApoE4 version of the protein actually affects Alzheimer's disease pathology in the brain. And so as we've alluded to, we think that genes affect behavior and cognition by changing the brain. And so how might this occur? So we have these genes, and these genes can code for things like proteins. And these proteins might include growth factors or peptide hormones or neurotransmitters like Dr. Kao mentioned, which help the cells communicate with each other. But they also can be little protein pieces that help make bigger things like cells. And these cells can go on to create neuronal structures and vasculature and help mediate immune responses. And so you can imagine how, if you had a mutation in a particular gene, that has the potential to change the protein that it codes and perhaps change the physiology or function of any of those downstream products. So I'm going to tell you about 4 types of genetic variation that naturally occurs in genes. So the first type are called short tandem repeats. And you probably haven't, but you may have also heard of these being referred to as variable number tandem repeats or microsatellites. And basically what these are short little pieces of nucleotides, or A's, C's, T's, and G's, the alphabet of our DNA, that occur in little segments, here represented as blue rectangles. And on different chromosomes, different people have different numbers of repeats. So one example is the CAG, this 3-nucleotide repeat that occurs in the Huntington gene, which causes Huntington's disease when it's expanded to a very large size. So another example of a type of genetic variation are copy number variants. And this is when there are big pieces of the genome that basically, instead of occurring at their normal 2 copies, you either get 3 copies or 4 copies or many copies more, or you get deletions. So you may only have 1 copy or you have no copies. And an example of that is the orexin 1 deletion that causes some forms of schizophrenia. And we next have a type of variation called insertion deletions. And this is basically an area in the genes where there's either a little section that's present or absent. And so one example is a serotonin transporter length polymorphism. It comes in a long size and a short size. And the short size has actually been associated with risk for anxiety and depression. And so finally, another type of variation that tends to be particularly dear to my heart because I do a lot of my studies using them, are single nucleotide polymorphisms, or SNPs. So these are, as the name you may have guessed, occur at a single nucleotide or base in our genome. And so in this example, on chromosome 1, on the top, you have a C here. But on the second chromosome, in the same position, you have a T. So these 2 different letters that are possible define that SNP. And typically, SNPs have just 2 different alleles that are polymorphic. So I'm going to talk more about SNPs later on when I'm talking about some of our large-scale genomic studies that we can conduct in people. So in addition to different types of genetic variation, we can also bin variation based on its frequency. So we have rare variants that are pretty uncommon and tend to be things that we think of kind of as mutations that cause disease or have very large risk effects conferring disease. And often these occur in families, or they're inherited in the big families like Aimee showed you. In addition, there are common variants, and these occur at a pretty high frequency. And because they occur at a pretty high frequency, they tend to be more risk-conferring rather than disease-causing, and they tend to have a small effect. And so in all 3 of the types of studies I'm going to be telling you about today, the fundamental question we're asking is actually a quite simple one. It's basically, what is the relationship between genetic variation and phenotypic variation? And phenotype is basically just a fancy word for, a trait, and in this case, a disease. So the first type of study I'll talk about are family-based linkage analysis. And so Dr. Gao talked about these a little bit in her presentation, and it's basically when we can collect families of people and then study the patterns of their genes. So just as an example, in this picture, oh yeah, and pay attention because there's going to be a little pop quiz. Okay, it's the only one though, I promise. Okay, so in this picture, this is a simple pedigree. The solid shapes represent people in this family that have the disease, and the open shapes represent people that do not have the disease. And in this example, and much like in linkage studies, what we do is we type all these markers across the genome, and then we look for patterns. And in this case, we could say, oh, we have a blue marker and a pink marker, and they mark 2 different chromosomes. So what we're looking for are basically parts of the genome that are occurring in all of the affected people in the family and not occurring in people that don't have the disease in the same family. So in this example, of the blue marker and the pink marker, which one would you be more interested in studying as potentially causing the disease in this family? Blue, yes, A+, okay. So you guys can go to sleep for the rest of the seminar because that was the only quiz. And so in these studies, as I mentioned, they tend to be rare mutations or variants that occur in relatively rare diseases. And as such, they have a very large effect size. So I'm going to tell you about one recent study that was published which identified a gene that causes FTD and ALS, or Lou Gehrig's disease. So as Amy mentioned, sometimes FTD and ALS can occur together. And so there were these families that clinicians and geneticists started collecting where both diseases were occurring in the same families. But what was interesting is sometimes people would just have ALS, sometimes people would just have FTD, and sometimes they would have both. And so it took geneticists quite a while, and eventually in 2006, we finally linked a region on chromosome 9 to, it's basically kind of like the blue gene, where it's segregated with the cases in the families, but we couldn't find the mutation. And it actually took about 5 years before we finally identified the mutation that causes disease in these families. And it turns out that that mutation is in the C9orf72 gene. This is basically a gene that we don't really know what it does. It doesn't even have a real name. This name stands for chromosome 9 open reading frame 72, which is a generic name that means this is something we think is a gene in our computers, and so we're just going to label it number 72 because maybe they found it as the 72nd one. What's more is this mutation is actually a hexanucleotide or 6-nucleotide repeat expansion. And so in normal-sized genes, we have about 20 repeats. But in the mutation, people actually carry upwards of 700 or over 1,000 repeats. And what's more, this expansion is actually not in a part of the gene that codes a protein, but it's actually in one of the things we call introns that some people would say, oh, that's like the garbage that you cut out before you make an RNA that's going to make a protein. But it turns out these probably also have a lot of important molecular functions as well. So here I'm just showing you one of the families that helped us map this mutation. This is the VSM20 family that was identified by Adam Boxer in our group. And through the mapping studies and subsequent studies of clinical populations, it looks like this mutation in the C9 gene actually is the most common genetic cause of FTD and ALS. And so for FTD, it contributes as much as 20% of familial and 16% of sporadic cases. And in ALS, it explains perhaps as much as 50% of familial cases and 20% of sporadic cases. And so since we're really interested in thinking about how genes affect brain structure, one simple thing we can do is look at the 3 different genes that most commonly cause FTD and ask the question, do they affect the brain in the same way? Well, it turns out it looks like the answer is no. So we have 3 genes here. These are 3 patients. One has a MAP-tau mutation, a progranulin mutation, and the new C9 mutation. And just to orient you, you may have seen MRIs before, but these are axial slices, so they're taken like this, cut through the brain. So on the top are the eyes, and then the bottom down here is the back of the brain. And these are basically cutting up. So in the tau mutation carriers, we see a pattern that looks relatively symmetrical atrophy in the frontal and temporal lobes of the brain. However, in progranulin mutation carriers, we actually often see this very striking asymmetric atrophy, where one side of the brain is just decimated, whereas the other side is relatively intact, at least in early stages of disease. And this can affect the frontal lobes and temporal lobes, but also can go into the back parietal lobes of the brain. And finally, in C9, we see yet another pattern of degeneration, where we see symmetrical but global cortical atrophy extending into basically all 4 lobes of the brain in some patients. The frontal lobes, the temporal lobes, as well as the parietal and occipital lobes. And it looks like in some patients we even see cerebellar atrophy as well. So this is just one example of how in some cases genes can have very profound and different effects on the brain anatomy. Okay, so the next type of study I'm going to tell you about are unrelated case control association analyses. So if you will bear with this complicated cartoon here, in these studies, basically we collect a lot of different unrelated people and their affected cases, and we compare them to unaffected controls. And what we're looking for are differences in frequencies of genetic variance between the 2 groups. So typically we would use this type of study if, for example, we wanted to do a study of ApoE in a group of people, and we wanted to see what's the difference in frequency of the ApoE4 allele in cases versus controls. But what do we do if we don't actually know what gene is causing the disease? Oh, and I should mention, for these types of studies, we typically have to use common variants because we need to be able to collect enough people that have them. And we do this typically with common diseases, so there are enough cases to collect as well. So we expect any of these findings in association studies to have relatively small effects. So if we don't know what gene we're looking for, we can utilize a type of study called a genome-wide association study, or GWAS. And this is actually what I did for my PhD, so that's why I have a fond place in my heart for these SNPs. And so basically what we do is we conduct an association study on markers across the entire genome. And typically it takes about 500,000 markers or more to accurately and nicely kind of catalog where we are in the genome. And so in these studies, I'm showing a little cartoon here. This is kind of how I think about SNPs in a GWAS. So rather than thinking, oh, I think a particular SNP is going to cause the disease I'm studying, we think rather that these SNPs are just kind of buoys or place markers that help us kind of get a sense for where we are in this genomic ocean. And as I mentioned, SNPs tend to have 2 alleles. And so we can look at the allele frequencies at each of these SNPs. And what we're looking for are frequency differences at a particular SNP or a subset of SNPs that occurs in cases compared to controls. And we're really just asking the question, what regions of the genome are shared in these unrelated people that all have the same disease? Because perhaps that genetic region has something to do with their risk for disease. So just as background for the next study I'm going to tell you about, I've talked a lot about ApoE4 as a major risk factor for Alzheimer's disease. So there are 3 different alleles in the ApoE gene, and these are potent risk factors for Alzheimer's disease. And As Dina mentioned, age is one of the main risk factors for Alzheimer's disease. And so we see actually with both a dose effect and an age effect that ApoE can increase the probability of getting Alzheimer's disease. So here is showing for women and men that have 2 copies of the E4 allele, for 1 copy, and for no copies. And basically you see as they get higher up on the graph and further along in age, their risk or probability of getting Alzheimer's disease increases. So this seems like a pretty big risk, but it turns out ApoE4 actually doesn't explain all of the cases of Alzheimer's disease. And in fact, there are many people that have no copies of the E4 allele and have Alzheimer's disease. And on the other hand, there are people with 2 copies of E4 and live very long and cognitively healthy, normal lives. So this suggests that there's maybe something else going on under the hood. And so researchers have gone out and basically tried to see, okay, well, what other genetic regions are in the genome are associated with risk for Alzheimer's disease. And so this is a study, or a GWAS, of late onset Alzheimer's disease. And so we think that genetic variation contributes about 60 to 80% of our risk for AD. And so these researchers sought to go after about over 2 million SNPs across the genome. And just to give you a sense of the size of these studies, this is the number of participants they looked at. So in the first stage of their analysis, they looked at over 8,000 cases. And in the second and third stages, they looked at basically over almost 40,000 people. And so these are huge, huge studies that require dozens and dozens of different research groups basically coming together and collaborating and sharing data, often across countries, to be able to create cohorts where you can identify one of 2.3 million SNPs that's actually at an increased frequency in people with Alzheimer's disease. And so luckily, they actually found some stuff. So in addition to replicating some of the findings that had been found before, they found 4 new gene candidates that contribute to risk for Alzheimer's disease. And these are just their names and their kind of functions. But mainly I wanted to draw your attention to the fact that their estimated risk is not that big. It's about 12% increased risk for any of the given SNPs in these genes. And so it's possible that you just need like 6 or 8 of these to contribute to your particular risk for Alzheimer's disease. But it's also possible that there's other types of genetic variation out there contributing. And so that brings us to our third type of study, or large-scale genomics. And basically, these types of studies are really complex analyses that we're trying to use to understand complex multigenic traits. And so here are pictures of just 2 of the sequencing machines that are out on the market today. And there are many others that exist, but at the end of the day, what all of them do they are really good at sequencing lots of DNA in parallel for, well, relatively lower costs than it used to be. And they take much less time than it used to take. And so, as you can imagine, with creating lots of data, you can get a sense, basically, by looking at an entire genome of all the common variants, but also of all these rare variants, or things that don't occur at very high frequencies and might even be unique to a given individual, but that we can basically start looking at in really large numbers of people to say, hey, are there differences between cases versus controls? And so, finding these rare variants, we can study both rare and common diseases. And we expect, since they're pretty rare, that they might have really large effects. So just to give you a little breakdown of next-gen sequencing, kind of our workflow tends to be we get genomic DNA from cases and controls. We do a lot of other stuff, and then we stick it on the machine, and we sequence the DNA. And then we take all of our sequence alignments and basically line them up and compare them to a reference, like the human genome. And if down here are all our controls, and in red are all the cases, we're basically looking for sequences that are different in cases, but not in controls. And in this case, I'm giving you a perfect example where all the cases of A's versus the controls that all have the G reference allele. But in reality, for these studies, we're often just looking for an enrichment of certain alleles in the cases compared to controls. So 2 studies came out together in the New England Journal of Medicine this year, and I'm going to tell you about one of them that was based in Iceland. And they basically did a deep sequencing project to identify a potent rare variant that contributes risk to late onset Alzheimer's disease. So they identified a SNP in the gene TREM2, which confers about 2 to 4-fold increased risk for Alzheimer's disease. However, this is a relatively uncommon SNP. In the Icelandic population, I think it occurred about less than 0.5% of the population. And in between the 2 studies, this one and another, it looks like it's probably a little bit less than 2% of the entire population of Europeans. TREM2 is an anti-inflammatory gene. And it typically serves as kind of a way to dampen inflammation. And so one thought is that in a normal function, it might help kind of protect against a lot of the inflammatory components of Alzheimer's disease. But perhaps in the mutation, they don't have this protection from the inflammation, and so they're more likely to get it. What was interesting is this graph here actually shows a subset of people that are unaffected. They're cognitively normal, they don't have Alzheimer's disease, but they're showing carriers of this mutation in red versus non-carriers in blue. And this scale is kind of backwards to me, but bigger scores mean worse performance. And it turns out that people that carry this, even if they're cognitively normal older adults, tend to do a little bit worse. And it gets worse and worse as they get older. And so this is one example of how human geneticists are really becoming very excited about this field of next-generation sequencing and starting these really big projects to try to find rare variants that contribute to disease. But the fact remains, we're still not very well equipped to deal with an entire genome. So if you like to watch programs like CSI, like I do, you might see them go into the lab and do a 10-minute experiment, and then they get a printout. And in the printout, it has this thing that says mutation. And so they know, like, oh, well, this is the suspect. He's guilty. But unfortunately, it doesn't work like that, actually, in the real world. I mean, besides the 10-minute window, we don't get this data from the computer. It actually just gives us all the letters, the A's, C's, T's, and G's, and then we have to try to sort them out. So as we're moving towards this really multi-locus, multiple gene approach of genetics, we're really going to have to amp up our statistical ability to really try to figure out how we can interpret this data better and apply it to the diseases that we're studying. And in addition, while we're doing this, we can identify new variants and try to investigate them in a biological way. And using animal models is a great way to do that. But ultimately, what we're going to need to do are also do these longitudinal studies really to assess the predictive validity of genetic variants. So what I mean by that is, for example, we could take a subset of people, genotype or sequence all of them, and then try to make predictions on their risk for different diseases. We would then follow them over time and basically see if we were right or not. And this is really, I think, one of the best and only ways that we're going to be able to move towards personalized medicine, where a patient can walk into a clinic, have their entire genome sequenced, and then have it be interpreted to them, and then given them told, basically, these are the risks for all these diseases you might get. Here are all of the therapeutic opportunities you could try to use to prevent getting that disease. And really, from the previous talks, you've learned that this might be particularly important for age-related diseases where, like neurodegeneration, you really have to kind of nip it in the bud before it even gets started. So at the Memory and Aging Center, we're starting to kind of embark on this new project of, can we map gene variation and expression onto differences in brain structure and function? And so we're trying to do all these neat things to collect data from, a bunch of SNPs across the genome, and then look at expression of all the genes and see if there's different patterns in their expression, and then overlay that onto kind of like atlases or maps of anatomical change of the brain. But as you can imagine, with these types of multimodal assessments, it's going to be really important to know exactly what we're studying. And so we're really going to have to be very careful with our phenotyping. And so, just as an example, I talk about case control studies. And so you can imagine how it would be really important to want to know that your cases are actually cases and that your controls are actually controls. And in addition, I actually had a study where a control was a case, but it didn't occur till later, so it got confusing. But anyway, so in addition to thinking about just case and control status being accurate, we might also want to think about subgrouping people that have the same kind of broad disease into more similar types of subgroups. So one example is Alzheimer's disease, which we typically think of as being very, very much predominated by memory deficits. But there are actually atypical forms or presentations of Alzheimer's disease that can also occur that tend to be more language dominated or visual spatial dominated. So perhaps in the case, instead of just lumping all of those people into a study, it might be better to actually parse them into 3 different groups and then study each one individually. And one way we can do that is with the use of biomarkers, or things like proteins that we can measure levels of in plasma or blood or cerebrospinal fluid. And we could also think about actually just using brain anatomic measurements as phenotypes. Because if we think that genes affect behavior through the brain, maybe it's easier if we just use the brain as the phenotype. And so some groups have actually started doing this and have been able to publish identify different genetic loci that they've identified in GWAS as contributing to, for example, size of hippocampus in normal people. So this could have really big implications considering the hippocampus is one of the parts of the brain most affected in Alzheimer's disease. And finally, I think it's equally important to think about how we can have very comprehensive behavioral assessments. And so you learned in your last lectures from our neuropsychologists about some of the different types of cognitive testing that we're doing to try to evaluate different types of cognition in patients and in healthy adults. And so with these very well-constructed tests, we can actually get a lot of information about the function of a brain, and sometimes even the function or size of a particular brain region. And it's certainly easier to administer a test than to try to get a patient to lie still in an MRI scanner for an hour. And so ultimately, it's our goal with using these different types of modalities to really be able to create a multimodal risk assessment for all of our patients. And of course, this is greatly complemented by us being able to then turn to our basic science neighbors, like Dr. Kao and Dubal, and have them kind of look into some of the molecular mechanisms underlying these things. And so just to come back full circle to this model that we saw earlier in Aimee's talk, I hope I've given you a better sense of how human genetics kind of fits into this cycle of studying human disease. And we see that not only from family studies, but also from these large, unbiased genome screens, we can start identifying genetic variants that might contribute risk to disease. And then we can take those variants and start studying them in things like worms and mice, and really trying to elucidate what the molecular mechanism is behind their risk-conferring status for disease. And an equally intriguing idea is basically to take genes that have been identified in mouse models that might contribute to things like aging or cognition, and basically use our new next-generation sequencing technologies to start screening those genes in people and see if we can identify rare or common variants in those genes that actually contribute to the same phenotype in people. And so just to sum up, I have 3 take-home points. First point is that studies of natural genetic variation in people can really help us identify genes that affect the brain. Second, Animal models can elucidate some of the biological mechanisms that might otherwise be quite difficult to study in people. And third, that human genetic studies really can benefit from the use of animal models and vice versa. So with that, I'd just like to thank you for listening. These are just, I didn't show any really of my data today, but these are just people that I work with and my funding sources. And actually, the three of us will all come up here and we'll be happy to address questions at this time. Thanks. The question was, I'm going to summarize. The question was, are people trying to identify environmental factors that might be contributing to Alzheimer's disease risk in addition to genetic factors, similarly to how environmental factors can affect risk for depression with the serotonin transporter length polymorphism? I would say broadly, yes. One example I can think of that's not directly related, and I'll have the two neurologists answer next, is this idea that traumatic stress or injury to the brain can actually start some molecular cascades of proteins that also can contribute to pathology or the things that go awry in the brain in Alzheimer's disease. And so one idea is basically to think about now that we should think about, previous history in terms of brain stresses or injuries that might be additional risk factors for disease. And another example is, I totally lost it. I'll let them answer and I'll come back when I remember. Okay.
So aging is the single greatest risk factor for Alzheimer's disease, but there are other non-genetic risk factors, and these include things like traumatic brain injury. It includes things like low socioeconomic status, a lower education level, trying to think of environmental exposures. I think there are some forms of heavy metal exposure that can increase your risk. Certainly there are pesticides that are associated with Parkinson's disease. So there clearly are what we would classically call environmental factors. But I think there's another layer to that question, because there is no pure environmental factor. I mean, our genetics are regulating our response to the environment as well. So there are certain people that are exposed to radiation that develop tumors. And then there are others who are exposed to the exact same radiation who are not exposed to tumors. And that is a combination of an environmental exposure and the genetic milieu that it's working upon. So in addition to risk factors that we've all talked about, there are also protective factors. Well, Dina talked about a great protective factor, but there are undoubtedly, and I'll give you a little hint that there's something big that's going to hit the news soon about protective factors that modulate our risk of aging and disease as well.
And
not from us. Which is why I can't tell you.
And I would add to that there is a really hot and developing field in all of science called epigenetics, and that is the study of modifications of the genes in the genome itself that can occur from the environment. And so we learned, I think almost a decade ago, that mouse moms that lick their pups and retrieve their pups in a healthy maternal way cause actually epigenetic or changes to the genomes of these pups that are good ones compared to those that were neglected. And that was at the level of the modification of the gene that was caused by the environment. And so this field of epigenetics on top of the genetics is really a hot developing field that number one, we're understanding the fundamentals of, but also beginning to apply that knowledge to understanding neurodegenerative disease. But we have a long way to go.
I remembered my example. I was just thinking of education, actually. The amount of education can also sometimes affect risk for Alzheimer's disease. So it can be argued whether or not that is a truly environmental or strictly environmental effect, but it certainly may play a protective role.
So attending this class tonight.
Yeah, you actually just reduced your risk 10% by joining. It's almost as good as a SNP. Yeah. The front row first, and then yellow. Oh. That's a great example. So the example from the audience was the idea, well, sometimes there are things like developmental or behavioral disorders that might arise that prevent people from being able to get formal education, but they can still pursue lots of educational or knowledge kind of enriching activities. And that's actually an excellent point, and something that people in our center are also starting to try to study more, are thinking about how these other changes in the brain related to development and behavior might actually help model the brain and make it protected or vulnerable to certain types of neurodegeneration as people get older.
There have been studies about how, in mice, about how an enriched environment can help brain size and brain maintenance. So the example would be, there's some mice in a cage and some genetically identical mice in another cage. And one set of mice get some toys to play with and the other set don't. And the ones with the toys, they overall do better, and they seem to do well on other measures. And so that starts to get to this other idea that's related to education and that I think Dr. Posehn or Dr. Boettcher introduced this idea of cognitive reserve as well, though. So I think there is clearly what you're talking about, which is that there's this modification that our genes make that then result in whether we graduate from the 12th grade or from the 20th grade. But if that extra 8 years of education occurs, there probably is a biological basis for why that protects the brain from disease. And it may have to, we don't know exactly what it is. It may be synaptic strengthening, it may be more growth factors for the neurons, but these are all these really, really interesting questions that we are very intent on hopefully someday answering.